Hello, we are now starting Chapter 4, which discusses cells. There are two major categories of cells, prokaryotic and eukaryotic. Prokaryotic cells are bacteria and archaea. If you go back to Chapter 1, you'll see that we discussed those two in the chapter. The other cells are eukaryotic. That refers to cells that have a nuclear bound membrane. In other words, they have a nucleus in which the DNA sits, most of the DNA. The cells that we are primarily interested in are going to be eukaryotic cells. This picture down here, it's a good picture to review later on because what it's showing you is prokaryotic cells do not have a lot of organelles. These are the two that are important. Eukaryotic cells have all of these plus these. So the only overlap is in this area. So prokaryotic cells and eukaryotic cells have a cell membrane that contains DNA. Now, this shows a picture of a generic bacteria. Bacteria are very small, very difficult to find underneath the microscope. There's good bacteria and there's bad bacteria, but for the most part, they don't have a lot of internal structures. They do not have a nucleus, but they do have DNA, and that DNA is in this region, which is called a nucleoid. They also have a rigid cell wall, which means that it's not flexible, bendable. Yeah, there's some bend, but it's not mushy like water. So here you can see the picture a little better. Here's the nucleoid, and that's where the chromosomes or DNA sit. Flagella. Well, there is one example of a flagella in the human, and I'll let you try figure out what it is. These pili are structures that the bacteria use to exchange genetic material or dock on different cells. So you can see that it does have some structures or organelles in it. Now, this is a very close magnification of a bacteria. And you can see they've tried to point out where the nucleoid is and some of the other structures that match the picture. So the pictures are nice, they're colored, and they are there to help you understand what they look like. Now this is a picture of a eukaryotic cell. This is what we are, an animal cell, and it's a generic picture of an animal cell. It's got all the organelles. Organelles are many organs that work inside the cell so that the cell can do its job. Your skin cells protect you. They prevent bacteria from entering. Your heart muscle cells contract to pump your blood around. Both very, very different from each other. And this picture looks nothing like that. But this is a good picture to help you as you try to study. I would print it, and you might want to take notes on it. So the first thing we're going to talk about is cytosol. Cytoplasm is pretty much the term that we use most of the time to talk about the jelly-like material. It's like Jell-O. And that's what's inside of the eukaryotic cell. And the organelles are included in the cytoplasm. Now, if we took away all of the organelles, you'd be left with a fluid type of material called cytosol. It has amino acids and other chemicals dissolved in it. Those aren't like organelles. Organelles you can see, you can move, you can do things with. But when you get hydrogen ions and other ions, those are at the chemical size, and you're not going to see a molecule. This picture and the next one are what they are in your textbook. This is another great picture of a generic, non-real cell, but it has put in place a better picture of what each of the organelles look like. Now we're going to be looking at everything in this picture. There are a few things in this picture that were not in that first graphic. And we're going to be talking about everything that you see here. Now this is a plant cell. Looks quite different. It's got a rigid cell wall. It's got something called a central vacuole. It's got chloroplasts for photosynthesis. So it's got different structures, but most of the structures are the same. They've got a nucleus, they have DNA, they have ribosomes. Everything else is the same. There's a few things that are different, and they will be pointed out as we go through the presentation. This is in your textbook. Now, the next thing we're going to talk about, we talked about the fluid inside. Now we're going to talk about what holds the fluid together. And it's a membrane that is very sort of liquid. They call it the fluid mosaic because that's what it looks like. It looks like a tile when somebody looks at it from a distance with a microscope, if they can get that close. The plasma membrane is critical. It is so important. It's what keeps the cell from its non-living surroundings. In other words, it protects the cell because inside of the plasma membrane are all the organelles that need to be doing a job. Now, I'm going to ask you to go back to chapter 2 and 3, phospholipid. It's a lipid, which is a fat, with a phosphate head. And it looks there's the phosphate head, and here's the tail. If you look right here, I just copied one of the layers. And notice that the head is pointed out. It is hydrophobic. It is hydrophilic. In other words, it likes water, whereas the tails are hydrophobic. So when you're looking at a cell, you're looking at the outside, that is watery, and the inside, or the cytoplasm, which is mostly water. And the only way that a cell membrane can exist is if it's got some fat. Our bodies are mostly water. The majority of our body is water. And in order for our cells to be able to function, if they were made of water, we wouldn't have a membrane that would stay together. So it's very important that we have this phospholipid present. So we've got the head that's pointing inside or outside the cell, and this one's pointing inside the cell. So the head likes water. It is pointing towards the watery side, whereas the tails do not like water. They're hydrophobic. They have a phobia about water. Hydro is water. Phobic is the phobia, so to say. The other thing that's being pointed out on this slide is that there are channels. And those channels are very important because it allows chemicals, nutrients, glucose, many different kinds of nutrients to enter the cell. If you remember the digestive system, you can go back and look at the nutrients that we have and that travel in and out of our body because we need them. Now, I left this slide blank so I could draw what I was just showing you. So here's a cell. It's got water on the inside, water on the outside. So fat and water do not mix. You already know that. So we have an environment where we've got to make the phospholipid bilayer. A bilayer means there's two layers of the phospholipid. It's got a head and two tails. If you remember in lab, you built a triglyceride. That was a lipid with three tails. This is very important as it helps maintain the integrity of a cell in our body. Now, the model that they use is described as the fluid mosaic model, and that just is a description of what the plasma membrane structure is. There are different kinds of cell membrane models, but the one that we use in this class is called the fluid mosaic model. And it is a bilayer of phospholipids. So here's our phospholipid so that you can see the chemical structure inside it. I know way back in chapter 2 and 3, it just seemed so foreign that these elements could combine together and start to make our body. Well, this is showing you the head and the tails. You've got the hydrophilic head. It likes water. But the tails are hydrophobic. They do not like water. Notice there's a kink. It's not saturated, if you so recall. Now, this picture is showing you what I've been drawing. That's outside the cell, inside the cell, and you can see the two layers. There's the head, there's the head, there's the tail, that's the tail. So there's two layers, bilayer. Now we're looking at the fluid mosaic model, and it looks like a mosaic, a tile, if you're looking at it from the outside. And you can see that there are proteins embedded in this membrane, and that's how certain chemicals get in and out of the cell. Some diffuse in and out through the membrane, like water and oxygen. They pass through a lot easier than some other chemicals that are in our body. Okay, end of part 1. We'll go on to part 2.